And now it's a pleasure to introduce to you Dr. Roger Hajjar. Dr. Roger Hajjar is one of our associate editors in JAC and is the director of Cardiovascular Research Center at Mount Sinai Health System. And he's going to talk about translational medicine from the protocol to the publications in a high-impact journal. Roger?
Thank you very much, Dr. Fuster. It's a real pleasure to be here today and to present some of my thoughts on translational medicine from protocol to the publication in a high impact journal. So, as an associate editor over the last number of years now, about 4 years, I've had this opportunity to really go through and look at some of the publications we've had in JAC and kind of put together some thoughts and examples of what are some of the interesting aspects of main JAC basic science or translational studies that we usually accept, and what it takes to actually get to the level of acceptance in the main JAC. So, in translational studies, just like in clinical studies, so human disease relevance is extremely important. So, when we're looking at a study, it's very important that it has human disease relevance in terms of clinical cardiology and something that will appeal to the general reader of JAC. I mean, you guys are all readers of JAC, hopefully, and you know the kinds of studies that you like to see clinically. And the translational and basic science and translational part also should fit in that same realm. Now, human tissues are very critical when we look at translational or basic, because this really validates the preclinical findings. Because many preclinical findings seem so off from everyday clinical work that we do as cardiologists that you really need to basically get the attention of the readers. The studies, even though they're translational, need to be have well-defined mechanism. So unlike many basic science journals, we don't really look for very specific mechanistic studies because that it belongs in another journal. But we really do make sure that the studies that are probing therapy or looking at the efficacy of a specific disease state have as their basis a very specific mechanism. Now, preclinical models in large animals and primates are well received, and they have to have a very specific protocol, and I'll get to that in a minute. So preclinical models of cardiovascular diseases are widespread, and some of the really best studies that we get are international studies from these large animal models. So I think this is something that I've noticed over the last 4 years. Now, IPS work, so inducible pluripotent stem cells and derivation of cardiac cells or vascular cells has really taken off in the last decade. And the modeling aspect of IPS work has become very important. And this is, again, another gateway of understanding the actual disease and testing ground. So, some of the important questions regarding human disease relevance, at least, is will this eventually impact clinical care? Now, unlike specific clinical studies that are published in JAC, we don't ask the question, is it going to impact clinical care immediately? But we do think about whether eventually this is going to have some impact in clinical care. We focus a lot on the novelty of the findings, which may lead to either new therapeutics or new diagnostics. So again, these studies that we like to have in the journal need to have a specific novelty. Now, unlike more basic or translational journals, or specifically translational journals, we don't look for novel methodologies, but sometimes this comes as a package in terms of the therapeutics and the methodology. So clearly, the novelty part is very important. Now, whenever you undertake, especially in large animal studies and preclinical models, you undertake a protocol. I'm going to come back to that in a second. You really need a robust statistical plan, and you need to get advice from experts before you even start the study. And this is really very much like a clinical trial. And then correlative studies that we do take in the main journal need to have state-of-the-art biomarker or genetic studies. So these are important studies that we consider, whether it's epidemiological studies or genetically based studies or pharmacogenetic studies, but they need to have really state-of-the-art um, base to them. Yeah, so in preclinical models, you should really set up your protocol as if you're running a clinical trial. It's important. So one of the most important things that we look for in these types of studies is that it's important to use the same endpoints as you would in a phase 1 or phase 1 or phase 2 clinical trial. And the statistical plan needs to be addressed upfront so that the study has enough power to reach these specific endpoints. So what do I mean by using the same endpoints? Now, you're going to say that many of the phase 1 or especially phase 2 clinical trials have clinical endpoints, and that's absolutely true. But I'll give you one example, and this comes from actually one of our studies many years ago. And this was looking at a gene therapy approach to reverse heart failure. And we focused on a specific endpoint, which is well validated in heart failure, and that's the LV systolic volume. And in these large animal studies, this is what was kind of our main endpoint, and it reached statistical significance in this case. Now, when we ran the actual clinical trial for this gene therapy circuit trial, we used exactly the same endpoint, LV and systolic volume. And the reason that was important, because we have known for many years that LV and systolic volume correlates very well with mortality in patients with heart failure. So bringing that biological marker back to the clinical scenario becomes very important. Now, another example is this left ventricular unloading with Impella in reperfusion, which we published recently from the Tufts group. And in this case, they're looking at infarct size using cardiac MR. And the large animal and pig studies that they do here specifically looks at this endpoint in their study. And then their clinical trial, which is ongoing right now, has CMR and infarct size as one of their endpoints. So you can see that how when we evaluate these large animal and preclinical studies, the endpoints are extremely important to look at. Now, as I mentioned, are the biological and organ-based endpoints predictors of survival and clinical outcomes? So these are very, very important. Now, the pronounced effects that we see in rodent models whenever you have a therapeutics are not reflected in large animal preclinical studies. And for that reason, it's going to be important to sometimes look at currently used medical therapies so that the model shows actual efficacy with this novel therapy. So you need to add sometimes regular-based medical treatment to the animal model. Now, of course, that makes it more complicated, but clinically, it has a lot more value. So in terms of the statistical plans, bridging studies from preclinical to clinical studies, it's really important when you're conducting your preclinical studies to keep in mind the clinical studies that you may end up with. So it's important to conduct simulations such as Monte Carlo to basically look at each efficacy endpoint that you're going to base your study on and make sure that these efficacy endpoints are based on sound literature or have strong input from experts in the field. Now, in terms of the results, make sure that you check for false positive rates in the statistical analysis. So this is done upfront so that you can power your study to preset biological differences, and you're able to basically get the magnitude of effect that drives the sample size. So these the statistical plan has really need to be basically thought out very carefully before your preclinical study is engaged. And that, again, makes it very important because we see many studies, many interesting studies go from a set statistical plan all the way to clinical trials, from preclinicals and clinical. Now, in the last number of years, we've seen a great interest in gene and cell therapy in many cardiovascular diseases, and that's really translated into preclinical setting where preclinical trials have been conducted before you get to the clinical or FDA allows you to go into the clinical trial. So we and other high-impact journals have published a number of articles focused on these studies. And then these biologics, such as stem cells or gene therapy, must demonstrate really significantly greater benefit than the current oral or other types of agents in order to become clinically relevant. So that's why, again, it's important to think about having a baseline treatment strategy that's what is used commonly in the clinics. These are, a number of studies looking at allogeneic stem cells promoting cardiac regeneration. This was done in animal models, and then the clinical trial was randomized allogeneic versus autologous mesenchymal stem cells in dilated cardiomyopathy. Interestingly, this is an example where sometimes a number of large animal use. So in the first study, there were only, I think, 12 to 13 animals, whereas in the randomized trial, there was 37. So sometimes the large animal studies are even fewer than actual clinical trials. Now, in biologics, in clinical trials, sometimes issues of safety come up. And that may be one of the biggest concerns and one of the biggest questions we have in the setting of a clinical trial. And the number of patients can be quite small, and the efficacy endpoint may be much less important. And one point in case was the human embryonic stem cell derived cardiac progenitor cells that was published recently in JACC, looking at patients with ischemic LV dysfunction. And in this case, there's been a lot of concern that human embryonic stem cells derived cardiomyocytes can proliferate nonstop and then create tumors within the heart. So this was a study done overseas. It was a French study by Philippe Menachet, which really answered that question about safety of using either iPS or embryonic stem cell derived cardiomyocytes. And for that reason, it was important to publish, even though there were only 6 total patients in the study. So iPS, the induced pluripotent stem cells, have really given us a nice window into clinical problems because these are in vitro model of cardiac disease that you can use for screening drugs and patient-specific cardiac therapy. They've really offered us new insights into a large number of inherited cardiomyopathies, such as long QT and arrhythmogenic right ventricular dysplasia, just to name a few, which will guide novel therapeutics. And with the combination of tissue engineering, they render these studies even more compelling. So just a quick summary, treat the preclinical studies really as clinical trials and develop them with solid foundation of statistical methods. And the biological endpoint should really correlate with clinical outcomes as you think about the overall preclinical setting. Thank you very much.